7,
8
8
module, 8
Okay.
modules.
Yeah, I will not suggest going them very quickly. I will suggest that you go one by one and make sure majority of the student can reproduce the thing on your slide, and then move to next one. How about that?
Okay, sure.
Yeah.
Perfect. That's sounds like a good plan. Yep. Thank you.
Yeah.
Yeah.
Anyway, those slides are shared, so when you introduce them, you can have a focus, and then people can go through the others. Yeah, if you cannot finish all those modules on your class. Yeah.
Okay.
Mm-hmm.
And maybe when I use these slides to teaching how to code, maybe I will use R to real practice with others.
Yeah. Sounds great.
Yeah, we having problem at line 27 onwards, like from read 10x folder from this.
Yes, I changed a little bit. Yeah, I will address that question.
Sure.
Okay.
Sure. Thank you.
So is this the right time to get started? Because it's already 3.
Yeah, I think you can start, and if some students are going to join us late, then they have the recording as well. So never mind. So please go ahead.
Yeah.
Okay, sure. So welcome back. In this class, I will teach you hand by hand, step by step to running the Seurat. So the first is to install the packages. I believe all of you already, I assume all of you already installed this. If you are not installing this package, please do so. And I will introduce the true source of our packages. And during this time, you can install the Seurat. Okay, the R package has 2 major sources. One is called bioconductor, and bioconductor is the biological related package pool, and a lot of package associated with the analyzing biological data, and those kind of package will publish into the bioconductor. And the second pool of the R package is called Crane. Actually, it's hosted by R itself. And there are too many packages in R, but not specific to the biological analysis data package. But the Seurat, they provide they host their package on this Crane. It's because this package is built in the S3 and S4 data format, which cannot accepted by the bioconductor. So they have to put the package into the this Crane package pool. So you just to install this package following this command, you can install this Seurat package directly from this package pools. And there are second way, there has another alternative way to download this package by using the using these div tools. Though the div tools is the tools for downloading the package directly from the GitHub, because the Satija, his lab maintaining the Seurat package on the GitHub, so you can directly download the package from the GitHub. And this version is under developed version, and that means this version is the latest. So don't worry about these 2 methods to download package. It's guaranteeing you download the latest 3.1 version of the Seurat. So let's get started. Before I'm going through this major download this this major section. For downloading the data, I will address one questions. Because some of you may not open this file. I think it's because some conflict of the download issue, some permission issue. So you can directly copy this command from my R script into the browser and copy it, enter. So it will give an instruction where you should download. And in the meantime, you should download the directory which you will perform major analysis in this directory. So please build a new folder for this file. Okay, you can save it and download it. And I believe it works now. If you have any questions, please interrupt me immediately, because this course is highly interactive, because I will lead you to run this Seurat package line by line. Okay, look at the download file. This is built in the R system. This command can help you to download the file from the website. So the first file we will download is called this PBMC3K filter gene BC matrix. That is derived from the 10x genomics. Actually, these two files, 1.3 and 1.5, both generated by the 10x, but with a different data format. The first is the first format is a folder. If you download this package and uncompressed this package, you will quickly see here. You will see the folder named like filter gene BC matrices, and you click in, you will find these three files, and I'm going to introduce what are those three files. So this is two different formats of the 10x genomics output. Here is the 10x folder and HDF5. Actually, in very restrictive terminology, HDF5 is the data structure. It's just provide a folder with these three different files in. And to the normal analysis, we usually use this general format, which is expression matrix. Sometimes we call this UMAP matrix. And each column in this matrix means the one cell. So you can see here, the cell 1 has this feature, cell 2 has this feature, and each row represents a gene. For example, the gene here is the gene name is Ep.4, and so on. And each value in this matrix are the raise number. Raise means the messenger RNA, and that means there are total 12 messenger RNA are captured under this cell of this gene. Okay, however, the single cell matrix have a very critical issue that called highly sparse. Yes, it's very sparse because up to the 98% of those value are 0. So you can, if you look at the whole matrix, you will find a lot of 0 inside it. So if we only store this matrices, it will occupy too many storage on your computer. And if you want to load this big matrix into the R, it will burn your memory. It will burn the RAM card. So instead, because these matrices are highly sparse, so there is a very smart way to store this kind of matrix. So these two formats of matrix is alternative way to install this kind of sparse matrix. And the first is a 10x folder, and next is the data format called HDF5. And under the 10x folder, there have 3 files. First is barcode. Well, the barcode indicates the cells itself. For example, the cell 1 maybe is indicated by this barcode, and cell 2 is indicated by a second barcode. So each barcode represents one individual cell. And the features is the name of gene. And the first is ensemble gene. Ensemble gene list. That is a database which stores a lot of genetic data, and they have their criterion to name the gene. And each gene in their database has their own format and document format. But this gene means they have a normal gene name and can be read by the biologist. It's called scRNA-Seq, the same as here. And the third column indicates the functionality of those genes. Its gene expression means. And here there is a major content of sparse matrix. It consists of the 3 columns. Actually, it's like a coordinate system because the first column is sorry, I should introduce what the header of it. So the first head of the first column is the number of rows in the matrix. So in this matrix, we have 31,000 rows, 31,057 rows in the matrix, and the second column means how many cells in this matrix. So in the matrix, it contains the 3,980 cells in this matrix, and this big number means the total number of reads. That means the sum all the values inside of this matrix and got this number of value. And you must guess what are the following of this content. The first is indicates the row number. That means in the 31,054 rows, and first column has one expression, and that expression value is 1. Okay, and the rest of things here are the similar and listed here. And in this format, it will dramatically reduce the storage space and easily can be read by the R. And this is a folder format. Another is HDF5. And this is typically, formally is the data format because it's only containing one file. If you download this file here, it's only have one file. It's called with extension name of H5. Does anyone has download this one?
Yeah, so this is a good point to take a pause. So students, are you able to get or install the packages, then download the filtered BC genes folder, and then this point H5 extension file?
Yes.
Okay. Cool.
I was not able to download I was able to download the H5 file. When I tried to extract it, it said that I didn't have the HDF5 plugin or something, so I installed that, and then it said it's not an H5 file.
I said you should download it one more time because I mentioned maybe it's because of the download the permission issue. You maybe just copy this link and open the browser and enter this download by this way in this way, so that you can smoothly reading this file.
Or it's just a suggestion. Is it possible that you can send us this email and this file on email? Or
sure,
just save it in the Dropbox link, and they can just, don't run that command. Just pick the command, like they have the save files, and they can just run it from directly.
Okay, sure. Yeah, sure.
Okay.
Definitely, yeah. After this class, I will send all the files into the Dropbox.
Yeah. So how large of the data is usually?
It's not large. It's few megabytes.
Yeah.
Okay. Then it will work. Yeah. I think. Yeah.
Okay.
But today's example data will be loaded from this matrices, 3 matrices. So it won't get involved into this this file. So
okay,
it's a problem.
Okay.
Okay. Let's continue. So the HDF5 is very like a directory. It's like a folder. It's like a container to contain the different slots. For example, the first clause is will store the matrices, and the barcode is corresponding to this barcode, the data corresponding to this matrix, and here is the row, the coordinates of the row, the coordinates of coordinates of the row, and here is the value inside of the matrix, and so on. You can find the detailed explanation at the, maybe you can find the detailed explanation at this website. I posted this under this under this figure. If you have more interest to explore what is HDF5, I suggest you can click this link and log in and to see what the truly HDF5 is. And okay, and then we need to read this folder. For example, in the Seurat, they have the built-in functions called read10x. And this function can read those folder which contains the 3 file. If you run this command, it will quickly reading this folder and read the data inside this folder. And if you do what I say just now, you will smoothly read in this file with extension H5. And I will run this code in the meantime because I already downloaded it. Okay, here I want to mention one thing. Before you're running this code, you should set a working directory because all the output and input will directly from this folder. So it's recommended so that in the following analysis, you won't provide absolutely file path for each file. You only give them a relative path. So you can read this folder. I'll just wait for a few seconds. And you can directly run this created Seurat object and by using this read folder. But before you're running this Seurat object, I want to show something here because it's very preliminary steps to explore the data. If you use this command, div, it will reveal the dimension of this folder of this matrix. Here you can see here we have 2,700 cells and these 30,000 genes inside of the matrices inside of the matrix, and then you need to create this Seurat object based on this matrix, and you direct to click here. Run this command, click. Oh, just ignore this warning. It's just switch name of the barcode of this barcode. So you already created the Seurat object here. Let's go back to the slides. Okay, the Seurat object is built-in under the S4 the system. And S4 is the object-oriented programming inside R. Because some of you major in the CS, you may think the R is not programming language. It's just statistics software. But inside R, they have their own object-oriented programming system. It's called S3 and S4. The difference, actually S3 and S4 are quite similar, but the S4 has more complex, formal, and restrict than S3. And S3 is suitable for the bigger projects such as the Seurat. Maybe it will store more than 2 or 3 gigabytes data inside of this object. So the S4 is recommended the format for this Seurat object. And because it's an object, so some structure inside of this object, and if you created this object, you can use this @ symbol to call each subunit inside of this object. For example, if you use @ to call this IC, and further, you can check the counts. The data counts means you already import the raw data into this object. And you can also recall the row counts by using this command. to prove that, I can run the dim function one more time to prove, okay, they're the same with original matrix. So through this way, you can calling back the row counts matrix. And here I want to emphasize two parameters under this function. One is called MIN cells, another is called MIN features. So what does that mean? I can explain this in the slides. So, excuse me. The minimum cells means if you actually these two functions is for the primary filtering, the primary quality control. It's the function of these two parameters is to filtering low quality genes and low quality cells. So how we define the low quality genes and low quality cells, we can define by this. The minimum cells means this gene should at least expressed by this number of cells. For example, if I assign 2 into, if assign 1 into this parameter, it will exclude the cells, it will exclude the genes which cannot be expressed by at least one cell. For example, if I set 1 here, it will exclude this gene, let me see, this gene and this gene, because this gene only expressed by this one cell. And this gene is only expressed by the cell 1, and this gene only expressed the cell 3. So these two genes cannot satisfy the threshold, so these two genes should be excluded. And the minimum feature means how many cells should at least this number of genes. So if we set the threshold as the 3, for example, in the cell 1, you can see inside this cell it already expressed 1, 2, 3, 4, and 5 genes. So we include this cell into the dataset, but instead we look at this this cell is only expressed 3 genes, 1, 2, 3, right? So we should exclude because it's not satisfies the threshold here. So basically, these two commands are used to do the primary quality control of the raw data. By default, the two parameters are 0. So if you you didn't want to, if you don't want to exclude any of gene or any of cell, you should keep this parameter as 0. So after we import the data matrix, the next things and created the thread object, the next thing we need to do is to do the real quality control here. So there are 2 matrices described here for the quality control. One is the percentage of mitochondrial gene. The reason why we care about this percentage of mitochondrial gene, the reason I already described previously, but I want to emphasize one more time. Because the percentage of mitochondrial gene can reflect the cell viability. If a cell contains a lot of mitochondrial genes, that means cell has been dying or it has died. So we don't want to include those cells in. So we should use this command first to calculate the percentage of mitochondrial gene in each cell, and after that. We can plot the feature, these 2 matrices. The first is the number of feature, and second the number of the percentage mitochondrial gene. And you can see here in the volume plot, each dot inside the plot represents 1 cell. And you can see these cells have very high express of mitochondrial gene. It's up to it's more than 20. So theoretically, we should exclude those genes. We should exclude those cells. But because I borrowed the default parameter from the Seurat official website, they use the 5 as the cutoff of how many percentage mitochondrial gene should exclude. I borrowed this threshold. So this command subset here means to take the subset of the cell which fit which fits those filtering criteria. And for the first figure is number of feature. It means the number of gene. Back to the previous slides. What is the number of gene? The number of gene is here. For example, the cell 1 have 5 genes expressed, so it has 5 features. So the number of feature is 5. The cell 2 has the 3 genes expression, so it has 3 genes. So the number of feature of cell 2 is 3. Okay, and this matrices evaluate how many genes are captured in the cell. Because this technology is based on the job-seq, so the median or the mean or median captures the genes inside of cell will be not very high. Maybe we can include those median genes in and exclude the extremely high feature cells or exclude the low feature cell. Here I want to pose the questions. Why we need to filter out those high or low number of feature? Why we fill out cell with high or low number of feature? Anyone can answer these questions?
So I have an idea. So maybe you want to filter out the dying cells because the cells dying adds noise that you don't want to deal with.
Yes, correct. Yeah, this is the purpose why we want to use this threshold. At least the number of features should greater than 200. That's perfect answer. So that remains one more question. Why we need to use this 2,500 to exclude the extremely high express cell? Who can give the answer or a quick guess? Okay, if no one have idea, I will tell you why. Because it's the job-seq, right? This technique is based on the droplets, which means, but there is assumption. Each droplet should contain one cell, and that cells should express around 1,000 genes. For try to imagine that thing. If one droplet capture more than one cell, what will happen? This feature will increase very obviously, right? Because one droplet capture 2 cells. So if one cell can be captured around 1,000 genes, that means the 2 cell at most captures the 2,000 genes, right? So those highly featured, so those cells with highly featured means they are not single cell. They are 2 cells or more cells. They are not the single droplets. It's a doublets or triplets. It's a triplets. So we want to remove those cells with highly featured. So am I clear?
Yuzhou, I like this point very much. I think even now in the single-cell sequencing technology development, those droplet one drop contain more than one cell, still a very challenging issue to be fixed. I do like this filtration here, and your explanation is pretty clear. Yeah.
Okay. So let's continue to move on. So after we filter out the low quality cells, and we will formally include all the play the real role cells into the following analysis. So the next step is normalization. The reason why we want to do the normalization is we want to remove the difference. Actually, it's a bias introduced by the experimental part. What that mean? That means during the sequencing this process, maybe you add this sample more into the sequencers, add that sample lower in that sequencers. So that will cause inequality of the sample size. So take the real example here. If I have the 3-cell got sequenced at here and captured those 6 genes in total, it's just a toy example. In the real, the dimension is more than this one. But take this example. The first cell captures the 29 reads in total, and the second cell captures the 32 features in total, and the third cell captures the 47 features in total, 47 reads in total. So they have a different sample size. They don't have the same baseline. So it's hard to compare which hair of each gene. For example, can you compare the RP1 gene in the cell 1 or cell 2, which have highly expressed? Because they don't have the same baseline, you cannot compare it. So you need to use some like the relative counts to compare those differences. So we divide each column, the sum of each column, the total number here. We divide the 12 by the 29 and got this 41%, and so on. You divide each value in the cell by the total number of column sum, and you can get the relative counts. And this is a basic idea of normalization. We need to make all the samples in the same line, a same baseline, so that makes all the cells can be comparable. So the default, the normalization method is called log normalization. Actually, it's very simple. It's just follows the method I just described. Divide this value by this total of number, and got this percentage of the expression value, and multiplied by this scale factor, say multiplied by this 10,000, and you got the idea how many numbers can get from the multiply from this multiplying the 41% and 10,000, and then do the log transformation to this through the multiplied matrix, and you can get the normalized data. It's very simple. And then after we removed those library size bias by using normalization, we need to get involved the further analysis. We want to pick up the highly variable genes from the all genes. Why we want to do that? Because remember, the raw data contains more than 30,000 genes. But if we want to include those all genes into analysis, it will first introduce a lot of noise into the such analysis. The second, it will give a very computational burden to your computer. It will run very slowly. So that's the reason why we want to only select the highly variable feature. And our algorithm for here is use VEST, is use the linear regression to weight the variance and mean expression here. The x-axis means the average expression for each cell. Each dot again represent one cell, and the y-axis is the standard deviation. It's the standardizing variance. So to form a linear relationship between those mean expression and highly variance, we can identify which cell has a feature of highly variable, have a very highly variable features. By default, the Seurat used the top 2,000 highly variable features. That means you can think in this way. This procedure is the first dimension reduction. It's used this by finding the highly variable genes. It decreased those highly dimension up to the 30,000 genes into those only 2,000. So I think it's kind of the first dimension reduction step. And if you're running this code, oh, sorry, I forgot I run this code simultaneously. My bad. I should run this one. If anyone run this code with any issue, just pop out and raise your hand. Raise your hand, so I can figure out what question you have. And take the subset. You can see here we use this criterion to fill out the low quality of cell and run this normalization. And here, very quickly, to find the highly variable genes, top 2000. And we want to visualize which gene we select by using this 3 command. And you can see here, I only showing the top 10 signature genes, which has a very highly variable, which has a highly variance. In another hand, by using those top 2000 genes, it will increase the possibility that we can find the right marker for the following analysis. And then after we got those 2000 genes, right, we cannot direct go to the further analysis because it's still, the dimension is still high. Although it's already decreased from 30,000 to the 2,000, but 2,000 is still a little bit high for computer and for us. So we need to further to do the dimension reduction. In this manner, we use PCA to do the first dimension reduction. And before the PCA, we should scale all the data. It's the standard process before running the PCA. The basic idea of scaling data is to shift all the data to the 0 center and scale by the variance. What does that mean? That means very, very simple. For the matrix here, to subtract this 12 by the mean of those rows and divide by the standard deviation of the strings, so that causes the scaling. So it's not very hard to understand. And here, inside of the scale data, it also provides another function. It's called regression out, unwanted variance. Because even though we use the percentage, the mitochondrial genes percentage to exclude those those low viability cells, but it still exists some variation between those mitochondrial genes, and we want to remove those variation. So we should, if you want to remove this variation, you should include these parameters in by using this vars to regress. It uses a linear regression to regress out the variation of percentage of mitochondrial gene. So in that case, we use the percentage of mitochondrial gene as the factor to regression out. But this procedure may take more time if you have more cell, because in this tutorial we only include the data set with no more than 3,000 cells. If you have more cells, for example, we have millions of cells, you cannot do this because it's really, really time-consuming procedures. So let's run in this command one more time. You can see here, only it has 3,000 genes. It takes so long time to run in the whole program. If it's not hard to imagine, if you have very large number of cells, it will burn your computer. And after we scale data and remove those unwanted bias arising from the mitochondrial genes, we want to run the PCA. Okay, it's the main theme here. And so what is PCA? In the very simple word is a combined effect of all those genes. Oh, sorry. For example, the PC1 and the PC2 here is not one genes. PC1, actually the PC means short for the principal components. And those pre-simple components depict the importance of a bunch of genes. Actually, it's all of a gene. And those genes will be ordered by the, by what that word? By those loading score. And those loading score can reveal the importance of each gene. And the PCA is actually a linear combination of those 2,000 top highly variable genes. If you want to know the more details of PCA, I highly recommend that you watch this video in the YouTube. And for me, because I have, because I'm biology background student, at the first time when I learned PCA is really struggling, but there are a lot of good resources online, and this YouTuber called StatQuest is really helpful to understand what is the basic statistic idea. And I highly recommend it if you you want to know what is PCA, you should look at this video, this here.
Yes, a quick comment. Basically, PCA is a linear regression and dimension reduction method. And like Tisney UMAP, they apply the nonlinear dimension reduction in their method. But usually, we can get started from the linear dimension reduction to see whether some very significant signal can be captured, and then move to the nonlinear ones. So I agree with Yuzhou. If you want to know more regarding PCA and other dimension reduction methods, there are a lot of information online. Yeah. So today, let's focus on the programming part. Yeah. Yuzhou, you can go ahead.
Okay. And after we run the PCA, actually, the PCA, this command consists of very complex mathematical theory, but in this command, it's only running with one command. Very simple, right? You just run PCA, and you can get immediately what's the output of PCA. And we want to, and then we want to, if you are a high-level student and you want to explore which gene play important role in the PC1 and which gene play important role in the PC2, I recommend you should use this command with the loadings. That is, visualize the loading score of PC1 regarding the genes, regarding of the genes. And just a quick introduce, if the genes with high loading score showing here, that means the gene play a very important role in this dimension. For example, in the PC1, I bet the CST3 should occupy at least one of these cluster. At least one of this cluster. And if you want to know the systematically, you want to know how the PCs, how all the genes participate in the PCs in the PC1, and I recommended you use this plus the plus heat map is regarding the PC1 and others PC2 to visualize each gene's importance inside of this PC. And inside of genes, just a quick introduction. Inside of genes, those genes are potentially the marker for the following analysis. So the take-home message is you should know this, at least this one command, run PCA. So let's try how to run this command. Run the PCA. Wait. Actually, in this command, there are a lot of parameters, but today, just to running by the default parameter, that's enough. It's cool, and run this visualization part as well. Can anyone follow me? Are you following me? I'm not sure whether I'm go through too quick or.
Any questions, undergrads?
Yeah, any questions so far? If no questions, I will move forward. And okay, so after we run the PCA, we will get another matrix. It's called, sorry, I didn't put this matrix into the slide, but you can imagine the gene cell matrix, the cell in the column, and the gene in the row, right? But after running the PC, we got a brand new matrix in which the PC, the principal components, they have a score. We can call it PC score. It's a PC score, and cell matrix is a brand new matrix. And by default, it will only contain the first 50 components of PC. So the whole matrix, the dimension of the whole matrix should, with 50 rows and 2,700 columns. And so after running the PCA, we got those pieces. We got a lot of pieces. It's a new dimension to interpret the data, but it's still high dimension. We still need to reduce those dimension again, because in our mind, we can only imagine something in 2 dimensions, in 3 dimensions. We cannot imagine how the 4 dimension looks like. So at least we should reduce all the dimension into the 3-dimension space or the 2-dimension space. So this step is to pick up the key feature of PCs. There are 3 methods for this selection. The first, we can, according to this heat map, I plot the first 15 pieces. You can identify this pattern, this diagonal pattern. They have a very strong signal here. PC1 strong, PC2 strong, but here not strong, right? Because it's blurred and not strong. And by eyeball check, we can see the first 7 PC is play a little bit. It's play an important role in the high dimension space. So we want to include, if we use this dimension heat map to select the number of pieces. By using this method, we should select the first 7 pieces. And there is not alternative way to select the pieces. For example, we use the elbow plot. It's showing the PC scores along with those pieces. And you can see the elbow. There is a turning point here. It's almost 7, right? 5, 6, 7. And here is a turning point. And again, it's proof we should select the first 7 PC to perform the following analysis. And the third one is more statistical significant because it's used statistical test. It's called JAX-STOR. Personally, I do not use this very often because it's time consuming, and although it gives those p-value regarding those PC, we should use the cutoff of 0.05 to select PC, but it's not reasonable, I think, because they didn't do the p-value adjustment, which is another statistical trick. So personally, I don't like this way, but I want to mention here this another way to select the PCs. The personal, I very highly recommend it to use this eyeball plot because it's very quick to check which PC you should include in, and it will take too much time. So I will run this code. First, plot dimension plot, it will take some time here. Okay, and plot the elbow plot. See the media, it will give the result immediately. But if I run this code, I won't run this code here. But if I run the code, I may not finish this workshop today. But if you have interest, you can run this code by yourself and check what's the result you got. If fortunately you will got exactly the same as me, showing the plot here. So after we select the first 7 pieces, and we want to visualize it, and we want to do the clustering based on these first 7 pieces, So we need to run the UMAP first. UMAP is another very hot, very hot dimension reduction method in the single cell analysis. By using this UMAP, you can see here, this is a UMAP result. You can get this very beautiful shape, topological shape of clusters. And if you're running this fan neighbors and fan clusters, you can quickly identify which clusters, you can quickly identify which group of cells belong to which clusters. Here, we identify the 9 clusters by using this 2 command. If you running this command and plot the dimension plots as here, and you will show, you will see the class information on the UMAP, this topological structure. And the reason why we use the UMAP, because it's really have a very good evolutionary effect. If we use PCA instead here, you can see here, it's ugly, right? The cluster not separate clearly. It's not very clear boundary between the different clusters. So this reason is again to prove we should use UMAP in the following analysis. And here if you have interest to know how the UMAP work, you should you can look at this video and learn how the UMAP work. And because this clustering funding, how to find those clusters, is based on the graph cluster. It's called the Levin algorithm. If you want to know more information about Levin, you can watch this video. It will teach you what is the Levin. But the workshop, the main idea of workshop is how to use this command. Actually, each command behind each command, there is a lot of mathematical theory, very complex. You cannot learn. It's very quick. Maybe you need to watch this video several times and also search another online materials for learning so that you can understand what is UMAP. But in the Seurat package, it's really well designed because you only run this simple command to help you accomplish and achieve the goal to reduce this dimension. And by using these 3 commands and plot those results, you can get the final result here, like this. And the final thing is the last part. What's the next step if we got those clusters? The short answer is, of course, we only know the number of clusters. We know, oh, okay, there is a different cluster, there is another, but do we know what the cell type is belong to? Because this data set is the PBMC, it's blood, it's a cell in your blood. So there is a lot of cell type in your blood. For example, T cell, the B cell, and the Y cell and red cell. You need to know the further genetic information to help you identify those clusters belong to which type of specific cell type. For example, do you know are those clusters belong to the B cell or those clusters belong to the T cell? You don't know. There is one way to know how to annotate those cell type is to identify the different expression genes regarding the different cell type. By using these fan markers, you can quickly know what the differential expression gene regarding the first clusters. The identity 1 means you choose the 1. The cluster is shown here. You want to identify what is the unique gene or the differential expression gene regarding this cell type. And if you got the result here, and I list the result here, and you can see the differential expression gene is RPS12 and other genes. Those genes are the differential expression genes regarding the cluster 1, and they are significant. The p-value and the p-address value showing the significance of those genes, and the log fault change is another way to know whether this gene are differentially expressed. The cutoff of the log4 change is 0.25. If this value greater than 0.25 or smaller than negative 2.5, that means this gene a differential expression gene. And the percentage means how many cells express this gene in the cluster 1. Take this gene example, RPS12 are 100% expressed in the cluster 1 here. And the PC2 means the rest of our cluster, because the comparison object is the rest of cluster except the cluster 1. We use the cluster 1 to compare the rest of 0, 2, 3 through 8. We compare the 1 and those rest of 8 clusters and to get those differential expression gene. So the %2 means how many cells expressed RPS12 genes in the rest of all the clusters. So the answer is 99.2%. So that's how to rate this table, how to rate this example table. And here is another way to test the different marker genes. For example, if we want to explore what is a differential expression gene of cluster 5, here is a blue here. What is the differential expression gene between this cluster and cluster 0 and 3, and show these two clusters? We can use these parameters. Use identify 1 means you want to give the 5 as source as the source cluster, and this identify 2 means give the 2 cluster as objective, which you should compare to. And by using these 2 parameters, you can get the cluster 5 differential expression gene regarding these 2 clusters. And the minimum percent is another parameters. It indicates how many cells at least, how many number of cells at least should express this gene. That means at least those cells, or at least those genes should expressed by the 25% of cells. Okay, and very interestingly, this group give one more function that calls
Hey,
yes.
We are almost run out of time.
Oh, okay.
Yeah. So I think we have to leave several minutes regarding the plan. And so can you quickly go? Yeah, if you have one more slide, you can.
Yeah, I have one more
next
slide.
time.
Okay.
Yeah. Can we quickly go to the last slide? Yeah, regarding next time. Yeah, the next one.
Okay, here because this script is based on the R script, the better way to show the results is to use Rmarkdown. The next time, one of the students called Sankun will introduce Rmarkdown, how to work with Rmarkdown and generate very beautiful website-based reports regarding single-cell RNA-seq analysis.
Yes, and Naren, this is a source code of the Rmarkdown for next Tuesday. And I already sent you an email including the slide and the example data you just mentioned today. And I highly suggest students involved in this group can check the Yuzhou slides again during this week. We have 1 week in between. Any questions, feel free to email me. I will forward it to Yuzhou to solve your questions. And Yuzhou will also be here next week. If you prefer further discussion of Seurat, Naren, you can forward the message to me. I can change my my content to have more examples regarding Seurat. Otherwise, we will go as we plan. How about that?
Okay, so there is one thing I would like to ask for the students. So
sure.
What if we can, for the next week session, So we can start by asking their progress, if they run the code for the Seurat, if they have some results to show, and if there is any error still, and if they are able to fix the read 10x error, that is good. They can just show us the results. We can just spend couple of minutes at the start of the meeting to make sure that they completely understand and run the code and they get the results. And then after that, we will go to the Rmarkdown lecture with your student.
Sounds perfect.
Okay.
Yeah, okay. I totally agree with that.
Okay. Yeah. Okay. Thank you then. Thank you so much. Yep.
Yeah, and I also want to mention Mirun is working with me. She is also very skillful in regarding single-cell RNA-seq data. So she's also a good resource to ask your questions. And
yes, no doubt. Yep.
Yeah. Okay.
Okay.
Any other questions before we closing this presentation? Okay. Thank you, Naren, and thank you everyone for joining us. And we can keep in touch and see you next Tuesday.
Sure. Thank you. See you next Tuesday at 1.
Yeah.
Thank you. Thank you so much.
Bye-bye, everyone.
Bye-bye. If you have any question, please email me.
Okay. It was a very nice presentation. It was very productive, I must say that. Thank you so much.
Thank you. Thank you.
Thank you for putting so much effort. Yeah. Bye. Bye.